Okay, I think we can start. So welcome everyone. Today we welcome Japanese delegation here. So welcome in set. There will be two talks, two short talks, but we should stick to the 30 minutes slot. So I will just leave Frank introduce the speaker for today, and also I think he will also tell how the day will be organized for the after this presentation. So Frank, please.
Thank you very much. So thank you very much everybody to be with us today. This Jeudi Marbec session take place in the framework of a new project that we have in Marbec, a new scientific project funded by the Occitanian Prefecture, based on Manila, on clam research, clam study of the population of Toh Lagoon. The project is described there on the first poster on the left. On the right, it's a participative science project. So you can see that we are linked with Maritime High School of SET in this framework. We have our colleague from Japan who supports the three other posters. We have Keiko Yamaguchi from Kirschel Company. Keiko, yes, Keiko Yamaguchi from Kirschel Company, and she have a poster, and she will explain this poster after this session. She managed a product to catalyze the Manila production in Japan. We have Shino Naito. She's professor in Maritime High School to develop economic project of the Kirschel Company. We have Sano-san, working on she is a professor in MIG University, and she works in genetic and epigenetic of clam. And after we have the two speakers, we will let you present yourself. They are from FRA, Fisheries Research Agency of Japan, and they work about marine science of bivalves and phytoplankton. So, Inoue-san, please, you start the presentation. Thank you very much. We just arrived from Manila Symposium, International Manila Symposium from in Arcachon, Monday, Tuesday, and Wednesday. So this morning we drive since 5 hours AM. So we have a big long road this morning, but now we have time to share. Thank you very much. Nariaki INOUE come two times, I think, in France. Is that your time or so? Two times. So he knows vaccine and
just use the keyboard.
You can use listen. Just apply. Just normally.
Good morning. So I am Nariaki INOUE, and I am come here Central Japan, near Tokyo. So today I talk about the presentation situation of fisheries and propagation techniques of Mania clam in Japan. So we will give two presentations today. So firstly, I speak, and next, Matsubara-san speak. So Mania clam production was in the 117,000 of ton per year before the 80s. But has been in continuous decline since then, and the catch level have been extremely low in recent years. So this is show 5 major asari clam harvesting area in Japan: Tokyo Bay, Mikawa Bay, Ise Bay, Suonada, and Nariaki Bay. So catches are decreasing in all major fishing areas. The situation is the same in Tokyo Bay. Multiple factors may be responsible for decrease of the minor clam resources. Maybe that vary from area to area and from year to year and life stage. To date, the exact reasons for the long-term decrease of large-scale resources are not known. So resources recovery have not been successful in any major fishing area. However, small scale in local level resources recovery have been successful. There are various factors that due to clam mortality in each area. However, common factor to many areas is wave and current. Many juvenile clams are found in the tidal flood, but after some growth, they are gone. In this case, wave and current may be the cause of the high mortality. So first, I talk about crash stone. Waves and current move bottom sand and form ripple on tidal flood. Generally, it is well known that clam are not found where sand rip area. Localized eddies form around the ripples, and these eddies can easily lift juveniles. Juveniles which are lifted by eddies are transported to unfortunatable and unsuitable environment and die. There are 2 ways to decrease mortality caused by transport due to wave and currents. 1, reduce the waves and currents near bottom. 2, stabilize the bottom sediment. For example, a bamboo fence is used for 1. Net cover effective for both, but installation and management are costly. In relation to 2, lying crushed stone on the bottom is effective. The crushed stone can be installed inexpensively over a large area. This is the construction material, low cost. Details are omitted. Clam juveniles of various sizes were placed in a tank lying crushed stone on bottom, in which the flow could be controlled and observed for juvenile move or not. Under the typhoon condition, for shell length less than 10 millimeter, crushed stone can be prevent almost all juveniles from moving. At size increase, the protective effect of crushed stone decrease. But however, in the case of shell length over 16 to this size, crushed stone can be prevent 40% juvenile from move. So demonstration tests have been conducted in Tokyo, Mikawa, and Ise Bay. So black line shows crushed stone, and red dotted line show control area. Juvenile density was higher in crushed stone area than both. Juvenile density was higher in crushed stone area than control area in both bays. So this is Funabashi case. This is an example of Funabashi, Tokyo Bay. Fishermen did the crushed stone line themselves. This is most important thing. Fishermen did crushed stone line themselves. For an area at the site could be installed in a few days with a small boat. red, red areas show shallower area. This figure shows changes in water depth before and after the laying of crushed stone, with a clear showing observed after laying, clear shallower observed after the laying. Comparison of Manila clam abundance inside and outside the crushed stone area. Pre-line survey 1 time and post-line survey 7 times. So black lines show crushed stone area and dotted lines show control area. This figure shows change in juvenile clam density inside and outside the crushed stone area. The density was clearly higher in the crushed stone area. In shallower coastal area, such as the fishing ground of Tokyo Bay, wind is the primary driver of current and waves. In the control area, in the control area decrease sharply after October. In Tokyo Bay, strong wind blow after autumn. It is indicated crushed stone protected juveniles for wind-driven currents. So this figure shows a distribution pattern of Manila clam density. Juvenile clam outside crushed stone had almost disappeared. Many remained, but however, many remained within the crushed stone area. Established 4 experimental group fills the mesh back with all crushed stone and 80% crushed stone and 20 hair shell, 5 crushed stone and 50 crushed stone and 50 hair shell, and all hair shells. Sorry, if you want to know detail, so detail, hair shell detail is oyster shell. Mesh bags were installed in June, monitoring regularly. So the larvae, the recruit from outside, grew 18 millimeter since shell length in only 4 months. 15 months later, each 4 bag samples sieved with this size. So in the groups with 0 to 50 hair shell, over 200 individuals were collected only a bag. On weight base, hair shell, 50% hair shell was the highest. So we did many trials. I don't describe the detail today, but I can say that mesh bag method can provide approximately 200 grams per small bag. And growth is faster than in normal tidal flood. Local population form meta population by recovering the resources of many local populations. And by further stronger the relationships of each local population, it may be possible to recover the Manila clam resources of the bay level. To achieve this, crushed stone and mesh back technique are effective. So this is all. Thank you.
I propose we follow, yes, and we make a question session just after you.
Okay.
What is your presentation? This one?
Yeah.
You have to this one.
これで、
Quelle bande? I just
Now, Tadashi Matsubara will talk about phytoplankton in Hiroshima Bay. Hiroshima Bay?
in Seto Inland Sea.
In Seto Inland Sea. So, Seto Inland Sea is a long sea, near 100 kilometers long and 20 kilometers large.
Yeah.
Yeah, something like that. And there is a lot of shellfish farming for oysters, but also for Manila clam. So please, Tadashi, thank you.
merci. こんにちは。My name is Tadashi Matsubara from FRA of Hiroshima branch. Thank you, Frank and Barry, introduced me and so long drive. We left our location at 8:30 a.m. 5:30 a.m. or more than 5-hour drive. Thank you very much. So my specialty is ecology of phytoplankton. So I study on the factors driving the changes of phytoplankton species composition in Seto Inland Sea. And recently I started the study on feeding environment of clams. So the contents of this presentation is reconstructed the poster contents. My poster at the Manila Clam International Manila Clam conference. So this is the second time my visit. I visited here 6 years ago. So I'm very glad to come here again.
Welcome.
Yeah. So I will start. In my talk today, I will talk about the food value of diatom genera, Skeletonema, Chaetoceros, and Leptocylindrus for Manila clam. If
just this one.
this
Ça marche.
Yes, thank you. Left figure shows the location of Seto Inland Sea, which is located in the western part of the Japan. And this is the largest enclosed sea area in Japan. And right figure shows the changes in the shellfish production, production of Manila clam. Like this, you can see that production of Manila clam has been decreasing since the mid-1980s. And this table shows the possible causes of the decrease in clam production in each area of Japan. So, in many areas, the coastal reclamation and over-harvesting were pointed out as the main cause. Among these, in Suonada, which is located in the western part of the Seto Inland Sea. Like this, the change of food phytoplankton species associated with oligotrophication was pointed out as a cause. So this right figure shows the changes in diatom composition in the Seto Inland Sea. Like this, during the high clam production period, you can see that genus Skeletonema dominant, quite dominant during this period. And after that, during the low clam production period, the dominance of Skeletonema decreased, and that of those of Chaetoceros and Leptocylindrus and Pseudonychia increased. So we thought that Skeletonema may be a good food for manual clam, and on the other hand, genus Chaetoceros, Pseudonychia, and Leptocylindrus may not be a good for clams. However, there were not scientific evidence for support this. So I started my study to determine the food value of diatom genera, Skeletonema, Chaetoceros, and Leptocylindrus for manual clam. At first, we conducted the analysis of nutritional components in Skeletonema, Chaetoceros, and Leptocylindrus, and subsequently feeding trials on the manual clam using the three diatoms was also conducted. So this slide shows the methods of analysis of nutritional component. We use the culture strains of Skeletonema marinoidoni complex, Chaetoceros neogracilae, and Leptocylindrus danicus. The culture of Skeletonema and Leptocylindrus was isolated from the Seto Inland Sea, and culture conditions are described the middle, and for the each diatoms in the late logarithmic growth phase, the water-soluble crude carbohydrates and water-soluble crude protein were quantified by the two methods. And this slide shows the methods of feeding trials. We conducted 3 20-day feeding trials on the manual clam using the 3 diatoms at 25 degrees. So this table shows the detail of each trials. And in trial 1, we use Skeletonema marinoidoni complex and Chaetoceros neogracilae as feed and conducted satiation feeding trials. And in trial 2 and 3, we use the Skeletonema and Leptocylindrus danicus restricted feeding. And this is the important, so please note, in trial 1 with satiation feeding, Skeletonema was, this is unexpectedly, Skeletonema was more actively consumed by clams than Chaetoceros. Therefore, the total feeding volume was larger for Skeletonema than Chaetoceros. So, therefore, in trial 2 and 3, the total feeding volume of Skeletonema and Leptocylindrus were unified. However, due to the low yields of Leptocylindrus, it was not possible to conduct satiating feeding. This slide shows the results of analysis of nutritional components. Upper figures show the crude carbohydrates concentration for each diatom, and lower one is the crude protein concentration for each diatom. You can see there were not big difference between the nutritional components of Skeletonema and Chaetoceros. However, it was found that the crude protein concentration of Leptocylindrus was very lower than other diatoms. So this slide shows the results of feeding trials. In trial 1, the growth of Manila clam fed with Skeletonema, described in red circle, is greater than that the growth of Manila clam Chaetoceros fed. The results, I think the results of trial 1 were considered to reflect the amount of food consumed. This is just a hypothesis. Chaetoceros may be difficult for clam to eat due to its long seta like a hair. So it is difficult to eat for clams. So in trial 2 and 3, the growth of Manila clam fed with Leptocylindrus, this is a blue square, is slower than skeleton fed. So the results of trial 2 and 3 were considered to reflect the low protein concentrations of Leptocylindrus. So this is a conclusion and future work. And conclusion, Skeletonema was considered a more efficient food source for Manila clam than other diatoms because it was nutritious and easy to eat. And future work, I think you think so. Feeding trials with unified total food volume for Skeletonema and Chaetoceros should also be conducted. I'm sorry. And this is what I want most to say through this presentation. To evaluate the nutritional value of phytoplankton for bivalves, it is necessary to consider not only the nutritional content, but also morphological characteristics related to ease of injection. It is very important, I think. Thank you. It's all.
Is there any question? Maybe we can start. If there is a question online. If there is no question online, maybe here in the room.
Sorry, I don't remember the names. Yes. I have a question for you. I just don't remember the name of the different diatoms, but there's one that seems pretty smaller than to others. And I was wondering, maybe it's not because of the nutritional, because they seem to have the same value of energy that they can give to the animal, but maybe it's in terms of the amount of energy the animal have to, to produce. Maybe it's not. 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 to expand towards the same amount of energy because it's really small, so that you fit a lot more of the individuals for the same amount of energy to. And I'm not sure it's
It's very difficult for me to answer. You said we could the same energy at the trial, each trial.
it's on your photos. There's three photos of
Yeah.
items, and one seems really small.
Yeah, at the kids cells.
Yes, and I was wondering maybe Chaetoceros is it's the same amount of carbohydrates and proteins. It's the same, but maybe they doesn't the energy the plant has to spend for to have the same amount because it's really small, so they have to filter a lot of water, so spend a lot of energy.
In our trials, for kids of negotiate, it is too small and too inject well. For maniacum is could not inject well because it is very small. So, so the results of trials one reflect the maniacum could not obtain the
It's too small.
cells from less than skeleton. But it is very important view to understand the feeding environment in natural water. There are many various types of phytoplankton that exist, so it is very important thing, I think.
So Chaetoceros is smaller, and there is cetas, but prevent palatability, ingestibility. The cetas, you're right, it could be a problem.
Maybe I think so, because we conducted using the larvae of oyster. You know, I will talk about. I talked about it. So, kids neograsside was not injected by the oyster larvae. Because I think because it was due to a long setup. But commercially sold kid cells loosen the setup due to the mass culture. Okay, so the morphology is different from the mass natural and soil species is different. So it is very important. Thank you.
Actually, I have a question for Nariaki INOUE. Actually, I was wondering when you how you select the site where you put all this material, and when you say that it's possible to scale it to the bay level. I mean, do you know the impact that this could have on other organisms, on the different currents and stuff like that? Is there studies like that?
Firstly, before rain, so simulation and current simulation with done. So if rain plus storm, too strong current not protected juvenile. So look for effective area. Firstly, so one question. So effective area, look for effective area and fishermen select here. Fisherman here. And more important thing, other kind of benthic animals. So, for example, Miyake's area protected area outside crust stone lying area outside, no animals. Only crust stone area animals. So we choose this area. This area is selected.
Are these areas are only dedicated for fishermen? They something like administrative area, and this territory is given to fishermen to exploit the 25 meters on 60 meter long. There is a state view of this administration. This part of this territory are only for fishermen.
Only for fishermen. So only for fishermen and fishing agencies, fishermen only use this here. This area.
Something like a concession, I think, in France.
Yes, sorry, but following this question, have you monitored the Banting community where you have thrown the crushed stone? Yeah. You monitored all the animals as banning clams. For several years, we monitor Banting animals. So 2 or 3 years later, only no monitor.
Only two or 3 years you conducted the monitor. Only 2 or 3 years.
After the materials has been put in the water.
Yeah.
And what have you seen as an impact on these Banting animals? 研究者がやるんだ。So for researchers study, only researcher check Banting animals. So after several years after, check fishermen.
So I understand that this part of the tidal flat is dedicated to fisheries, and due to typhoon, there is a large erosion of this area, and your protocol is made to compensate the loss for fisheries. That's it. The protocol, the methodology you develop with Spochton is done to compensate the erosion, the natural erosion due to typhoon. So my question is, what is the frequency, the evolution of the typhoon frequency in Japan? What is how many typhoons you record by a year, and are there any evolution of this frequency for typhoon?
Depending on years, maybe
higher.
10.
10 typhoons by year. And are there any evolution of the number of the typhoons due to the climate change?
Well, we don't know.
Are there any by year?
Here.
大体ね、number of typhoon is increased due to the global warming, but the number of typhoon hit the Japan is not is
decrease.
they go to the other area, but number is increased.
So depending on location, for example, Miyake, many typhoon. So but 3 years or more effective limestone heat.
Actually, we have a less typhoon before.
Yeah, really.
But total number is increased. But we have less diatom.
Okay.
Maybe in 10 years. Listen. Now.
Thank you very much for your both presentation. I have one question for you, Tadashi. We have observed the same pattern as in Chaetoceros Sea regarding phytoplankton because of oligotrophication process. Skeletonema has declined, Chaetoceros has increased. And recently we saw that every diatom decreased, and with warming, we are watching more and more often Cylindrotheca. It is a benthic diatom which can be released in the water column. Have you observed the same pattern about this species?
No, because the inland sea is deeper than your Philto Laguna. I think the increase of Cylindrotheca, which is a benthic diatom, is due to the tolerance, water tolerance. Right. With related to the bottom due to eutrophication, and so I think the benthic diatom could obtain the enough right to growth. So situation is so different due to the deficit of our field.
Thank you.
So if we have time, we can take a few minutes. If you can stay here and you can have a look on the different posters. Matsumi Sano-san is there to explain his work about the assessment of feeding of juvenile Manila clam using gene expression. But I don't know if there is anybody in the room specialist on gene expression, but
actually there's one question online.
There is one question online,
which is really general, and actually Arif is interested in how oligotrophication can happen in Chaetoceros.
There is a question
One question
for you.
Actually, someone is asking how the oligotrophication can happen in Chaetoceros.
In Chaetoceros, what causes the oligotrophication?
Yeah.
It is usually the pollution control. The very severe pollution control was conducted in this along the Chaetoceros Sea. So the amount of amount of total nitrogen and total phosphorus was domestically decreased. So in these 30 years, so it is a cause for the oligotrophication of Chaetoceros Sea.
Okay, so yeah, so I guess we can first thank you again, and thanks to the people online, and we meet hopefully next week, because actually the talk has been cancelled, so we are looking for someone to give a talk, preferentially in Montpellier, and so see you next week. Thank you.
Thank you.